Okay, let's go on to topic 2. And the topic 2 is labeled as MRSA. Now, this is a mouthful. It's methicillin-resistant Staphylococcus aureus. Staph aureus is a form of bacteria. It's something that we have on our skin or around our body all the time. Okay, but what MRSA is it's a bacteria that has grown resistance to antibodies, and it's a very big concern because we all take antibodies for everything, and eventually through time various bacteria will grow tolerant of that. We've all heard this, and then we have to find a different type of remedy for whatever ails us. Okay, and we've all experienced it where, you know, a doctor gives us something and it doesn't work, and it says, and you got to go back to the doctor. That's why I say come back in 7 days if you don't feel better, and then they try a little bit more strong type of antibiotic that will help you get better. Okay. Anyway, so this is what we're going to be talking about. It ultimately wraps itself around the concept of evolution. So we're going to look into that briefly. I'm just going to skim through that because we're not going down the rabbit hole very far with evolution. There are classes strictly on that. If you're interested, you're more than welcome to take those classes as you move along. But otherwise, I'm just going to give you the nuts and bolts of probably concepts you already have heard and understand. So, of course, when it comes to evolution, we just want to make sure that we understand that it's Charles Darwin who came up with this concept. The concept's been around for thousands of years, but it is a discipline, a form of thought in relation to science of how organisms change or adapt to the environment. And that was his observations were. And as all scientists, we are all scientists. He just happens to write things down and observe things and write things down in catalogs and so on and so forth. You know, a lot of us that work in the kitchen, we're scientists. Sometimes we create concoctions and hope to goodness that it tastes good. You know, if it does, some of us write down the recipe. So again, in actuality, we're a scientist ourselves. Anyway, so this is just a brief history. You don't have to know this. I just want you to get an appreciation for who Darwin is. He was this, again, around the middle of the 1800s. Okay, so there are a lot of people that did some magical things in the 1800s. But he's from Cambridge. He was a wealthy individual, upper class. And this image down below is his path, because he took this particular ship. If I can find my mouse, it disappeared on me. There it is. He took this particular ship, a very nice looking ship for the time. Okay, the HMS Beagle. And then I think on the next slide I have it. Yeah, so when he went to the Galapagos, these are the adventures that he took, and that's where he made a lot of his observations. He spent a couple of years down there and noticed a few things. Now, what we have here is an image of finches, and these are called Darwin's finches. It's the finches that, and the variations of the bird and its environment that made him start to think about things, about evolution. And specifically, you'll notice that there's a variation in beak, okay? And then also you have a variation in color of the feathers. So there was some birds, some finches of one type that flew, whether by storm or whatever, many years ago, and landed on the Galapagos. And then over time, you know, they adapted for survival. Okay, so I would think that, as an example, the lighter colored feathered finches probably live closer to a beach where there's white sand. They blend in. Okay, if you'll notice the beaks, that they are probably more prone to getting insects or digging, and maybe the beaks that are a little bit stronger, shorter, and with greater musculature in the jaw might be on the beach line where there's foliage for seeds and such. And then your darker colored birds are in a different environment, maybe more interior where there's more jungle and shade, and they blend in from predation. It is about staying alive and what helps you stay alive and what helps you adapt to the environment for longevity. Okay. Anyway, these are known as Darwin's finches, so keep that in mind. A couple of things I want to pass along to you is that Um, Darwin was not ready to enlighten the world when he discovered this and realized this is something. He thinks he's got something here. But he knew that the world wasn't ready because they had different ideas about evolution or how people changed. As an example, it was perceived back in the 1800s and earlier that if as you grew up you became a great three-point shooter in basketball and you could hit long shots and so on, that if you had children, then your children would have that same gift. And that, of course, is not the case. Your child may inherit your musculature and all the necessities to become a great three-point shooter, eye acuity, and being able to see through distance and so on. So he held back on this, and so didn't think that the world was ready. So what his thoughts were is that favorable variations would tend to be preserved and unfavorable ones destroyed, which is considered descent with modification or natural selection. Okay, so what it means by favorable variation, so anything that helps you adapt to your environment is a good thing. Okay, so us having hands and being able to walk and have fingers for dexterity to grab tools and such, these are good things. They will not disappear on us because it works for us. It enables us to survive. Okay, and if we had traits that did not really help us, would eventually go away. Or if it did not enable that organism to survive in the environment, it would disappear. Okay, it would die off, and then those genes die off with it. Okay. Descent with modification is simply passing traits from parent to offspring. Okay, so if you've got blue eyes, then there's a very good chance that your children will have blue eyes. That may not necessarily be the case, as we will learn in genetics, that it depends on what your partner has as well. But ultimately, whatever you have, part of what you are gets passed along to your children via DNA and the genes that are within DNA. Okay? There's also another gentleman named Alfred Wallace. Just wanted to talk about him real quick. He was another naturalist that made quite a few observations, but this was out in the Indo-Pacific. Okay, he was not necessarily as wealthy as Darwin was, but he was also of the same mindset. And what is pretty cool about this gentleman and Darwin is that here in 1858, Darwin was he gave partial credit to Wallace for coming up with the same concepts as he did. Okay, and one of those concepts again is in every generation, weaker individuals will die, and those with the fittest variations will remain and reproduce. And as a result, the species will become better adapted to its environment, survival of the fittest. We've heard that. So it just means that, as an example, ducks. Way back in the day, ducks didn't necessarily have webbed feet. Okay, so if they're in the water and they didn't have webbed feet, as we know as swimmers, that if you close your fingers, you can get a little bit better velocity in the water. So those that did not have webbed feet would fall victim to prey because they would be last. In comparison to those ducks that apparently there might have been a mutation where some ducks have webbed toes or webbed feet, gave them a little bit of extra edge, become faster than the other ducks, and survive the predation event. Okay, since they survive and they have the genes for that particular trait, then when they had little ducklings, then those ducks have webbed feet, and you get the idea over and over. Now we know ducks that have webbed feet. Just like we have dogs. There are some dogs that have webbed toes that are water dogs. There's some that are not. Okay, that is a trait. And that assists in survival. So those that do not have webbed feet with ducks, as an example, no longer exist. They fell to predation. Hence, their genes that produced that particular trait died off as well. Okay, so that's what that means. And this is an interesting little concept. I like showing this. So here we've got an environment of different colors. We've got the beetle M&Ms. So here we've got a population of M&Ms. You've got all different colors, and you've got the environment that also has all different colors. So all these M&Ms are distributed throughout this particular environment. You don't see them, okay? But through time, the environment changes. So we don't see, what don't we see? We do not see the orange, okay? So it looks like the orange grasses have disappeared, okay? So that means that there's going to be a change in the environment. What else has changed? Green is there, blue, brown is there, red is there. Yep, so orange. Because orange, that means they have nowhere to hide. They are more visible. They're going to get eaten. So now that gene for red is gone, or excuse me, orange is gone. Okay, more time. Now the environment has changed again. It looks like red no longer exists. That means the red beetle, or M&M, has nowhere to hide. It will stand out, and it's gone forever. So the gene that makes a red-coated M&M beetle no longer exists in the gene pool. And likewise, over time, now yellow. Okay, you get the idea. So now all that's left, that was a nice robust community of different color M&Ms, is now down to 3, green, blue, and brown. Okay, so change in allele frequency changes the gene pool. So what is a gene pool? Total number or collection of alleles in a population. What is an allele? Allele is, we haven't gotten to it yet, but just so you know, an allele is a variation of a gene. As an example, we all carry the genes for eye color, right? Mom, you, Dad, everybody's got a gene for eye color. But there are alleles. One may have the gene for eye color that codes for green eyes. There's another, someone, say Dad has that. Then Mom has the gene for eye color as well, obviously, but maybe it is brown. Okay, so those are alleles. Allele is nothing more than a variation of a gene. So here's just in conclusion, we have our initial gene pool of 6 robust colors in the environment of which they live in. You have a change in the environment because the environment is always changing. And then we have a change in the gene pool. Okay, so we start with one, we end up with another. All right, natural selection. They love to use giraffes, so I got a lot of giraffes. But again, this is the same thing, and I want to move on because I don't want to make this a super long video. Natural selection can only take place if there's a variation within a population. What does that mean? That means with these giraffes here, if there's natural selection, that means that if the food source is up high, we have both a long-necked giraffe and a short-necked giraffe. At one time, giraffes had short necks. You don't see giraffes with short necks anymore because that gene pool died out many years ago. Okay, something happened where the environment changed and giraffes that had long necks were able to get to the food source easier than those that did not. Those that could not get to the food source died. Those that had long necks survived and were able to reproduce. They carried the gene with long necks, and that trait survived on and on and on. So individuals with good traits for their environment reproduce and pass on those traits. Okay. Individuals with poor traits for their environment die before they can reproduce. So all that should make sense to you. Okay. And a lot of this is similar things, so I'm not going to beat you up over the head with that. I highly encourage you watching Evidence for Evolution. It's pretty cool. It involves fossils, anatomy, biogeography, and molecular biology. It's a very short video, shorter than mine. I can skip. These are just, again, redundancy of important points about evolution. I've pretty much already talked about everything I need to. Now, how does it, what is this all about when it comes to MRSA, okay, or Staph aureus and antibodies? Well, obviously, what happens is natural selection. Those bacteria that can survive the onslaught of antibodies will survive and carry on. Those that do not or succumb to the antibody, which is what we want, they die, okay? But over time, these bacteria are going to get more resistant to the antibodies that we developed. And of course, we developed these back in the 40s, so they haven't been around that long. Okay, but as it says, some of the antibodies that we create are useless because of this resistance, this evolution, if you will. Okay, and so I got a lot of examples the book uses. Okay, so as you can understand, here's a variety of bacteria: non-resistant bacteria, antibiotic resistant strain, gray and yellow. And you can see that those that are not resistant are going to succumb to the antibiotics. Those that are resistant will thrive, and then of course they will survive and carry on. So that means that we have to have a different antibiotic, because you can see these yellow bacteria are still there. You have to go back to the doctor in 7 days. And of course, that's what we're talking about with antibiotic resistance and MRSA. So make sure you know what MRSA means. Methicillin-resistant Staphylococcus aureus. I always have to look at it to remember, so good luck on that one. Okay, antibiotics. That is just strictly an antimicrobial drug. Okay, it's something that we can put into our bodies. We found that certain bacteria are defenseless against these antibiotics. Okay, and then these particular molecules. We actually do this in lab and look for the zone of inhibition with E. coli. And then there are some that are resistant to it. So antibodies we put into our bodies. Of course, our bodies naturally produce antibodies, but then there's some that we take antibiotics to give ourselves a better boost to overcome the pathogen that's in our bodies, whatever bacterial infection we may have. So here's something that's kind of cool, is how do antibiotics work against bacteria. So tetracycline, some of y'all have taken tetracycline. That interferes with bacterial ribosomes. So if it affects the ribosomes, that means you have a problem with the bacteria replicating DNA. If it can't replicate, it dies. So that's kind of cool. So penicillin disrupts the formation of the cell wall. If the bacteria doesn't have a cell wall, it can't form. It dies. Ciprofloxacin, okay, that destroys the chromosomal makeup. Okay, so what happens is that if it messes up the DNA, that means that it can't form, it can't reproduce, it can't create proteins, it just can't function. Okay, and so the bacteria dies. And then cytoplasmic enzymes, or sulfa drugs. So this is what affects, I guess, certain enzymes that have been produced. And I suppose it depends on what stage of the disease or infection you have when they dictate what kind of antibody is good for you. Are you allergic to anything? How severe is the infection? So on and so forth. So this is why doctors get paid big bucks to understand all that. Anyway, I thought that's kind of cool. So antibiotic resistance, again, another illustration. You get the antibody, try to kill off all the bacteria. Some survive. The ones that survive reproduce. Those that can't die. And then we have to go back and visit the doctor again. And then a quick side note when it comes to understanding bacteria. Bacteria are prokaryotes. Okay, they don't fall among the plants, animals, and fungi kingdom. Those are eukaryotic cells. But the prokaryotes can affect plants, animals, and fungi. Okay, they're very, very small. Prokaryotes are the first organisms or groups of life on Earth. Okay, like cyanobacteria, and that they also are the ones that helped change the atmosphere many, many millions of years ago. Okay. So there's two domains of bacteria. You've got domain bacteria, domain archaea. Archaea, archaic, Biggest thing about these, they're also called extremophiles. You may have heard that in your biology classes before. So domain archaea, these are bacteria that live in extreme environments, acidic, very hot, very cold areas. This is what gives xenobiologists, those that study life on other planets, they get all excited because it wasn't all that long ago that we realized that life can exist miles under the ocean by hydrothermal vents in very hot areas, very acidic conditions. We thought that life could not exist and does. Okay, and then domain bacteria is basically everything else, all bacteria found everywhere. Some are cause diseases, some are beneficial. We have bacteria in our bodies. Okay, your gut is filled with them. That's one thing. If y'all have been given a Z-Pack, you're deathly ill. Doctor doesn't know what's wrong with you. He gives you a Z-Pack. It wipes out everything. Oh, sure, you can go back to work, but you're not feeling the greatest because your gut has just been, your body's been destroyed of that good bacteria. It took the bad stuff out as well. So now you got to replenish your body with good bacteria that helps break down foodstuffs in you. So you need to have good bacteria within you, but that's what a Z-Pack does. It just knocks everything out. If they don't know what's wrong, so here, take this, and that'll take care of it. Make sure you know some of the anatomy of a prokaryote. They have cell walls. Okay, a lot of them have what's called flagella that helps them move around. Most have a capsule, which gives them protection. Okay, from the environment. An endospore. And then how do they produce? They just split in half, or binary fission. So when it comes to the growth of bacteria, they grow exponentially. If there's plenty of food, plenty of space, they will just keep splitting over and over and over again. A lot of the information that we have acquired on genetics is based on bacteria, because you can make a change to the gene pool and find out within 24 hours what happened, versus a human, which is a lifetime. Okay, so bacteria is very good to study when it comes to genetics. Make sure you know the different shapes. We've got cocci, bacilli, and spiral. So there's different shapes of bacteria. All right. So there's several of them, but these are the main components: the spiral, spirilla. Hope you can remember that. Bacilli, there's lots of I's and L's, so that's rod-shaped. And then cocci, C's and O's, they're round, so that gives you the circular spherical shape. Let's see if there's anything else that's exciting that we need to know. Just what bacteria can do, okay? We use bacteria in sewage treatments, bioremediation, your toilet, if you're on a septic tank, you put bacteria to break down all the sludge. Okay. Pathogens, that's known as a bacteria that causes illness. You got Staph aureus that can form, again, it's all over, like in your lockers. Anthrax, or Bacillus anthracis. Salmonella, you find in restaurants, that's why the health department always visits your restaurants to make sure you're not spreading diseases. Lyme disease, which comes from ticks, that's a bacteria. I talked about that. And then lastly, I think this is near the end, is how bacteria can transfer DNA, or how do they reproduce asexually. There's four types: transformation, transduction, conjugation, and plasmid transfer. Okay, so let's take a look at them real quick. So transformation, you got foreign DNA. Okay, so then that gets embedded within the bacterial cell wall. It's able to get in and integrate within the chromosome. And then there's a transformation where then that new DNA is found within the chromosomal makeup. Okay, that's called transformation. Okay, so dead bacteria can release DNA into the environment. So that's what this foreign DNA is. It could be something else. But now this bacterial cell that's over here, transformed bacteria, is now different than what it was right here. Okay, so it has, quote, evolved, incorporated some other DNA that it didn't have originally. So that's one form. Bacteriophages is a virus that infects your bacteria. Okay, so here's a bacteriophage, looks like a little spaceship or capsule. And the virus is a type of virus that has its DNA. There's a battle or an argument within the science community whether they're actually dead or alive. They cannot reproduce on their own, so what they do is they go ahead and attach to a bacteria, inject it, the DNA, which then incorporates itself into the DNA of the chromosome and utilizes the components and then creates its own new chromosome. And then in conjugation, we have these pili, where they form connections, okay? And then conjugation is where they actually merge together, open up their cell walls, and transfer DNA, okay? And the last one is plasmid. Can't forget the plasmid. This is going to be important when we talk about gene therapies. Okay, so just to let you know what a plasmid is, it is a piece of DNA that has genes, but it is not the complete DNA or chromosome makeup as the main chromosome. Okay, so you got the bacterial chromosome, and then you have what's called the plasmid. Okay, it is just some of the genes, not all of the genes. But anyway, that gets replicated, okay, and as it forms, as the bacteria splits, it gets replicated as well. So it's that plasmid where we can inject other genes that then get taken up by other bacteria. Okay. So as it says, plasmids can replicate independently of the bacterial chromosome. Okay, that is all for this. This is a longer video. I apologize, but it's lengthy. But definitely work on the Pearson assignment, and it's got some good questions and thorough information for you. And make sure you do read do the reading assignments. Okay, we'll see you on the next one.